All right, so I think we might get started. It's a good turnout today. Thank you, everybody. I'll begin by acknowledging that we are all here today on Indigenous territory, land of the Wurundjeri people of the Kulin Nation. So I'd like to start by paying my respects to the elders past and present, and welcome any Indigenous people who may have joined us here today. It gives me great pleasure to introduce to you Dr. Darren Creek, who will be pretty well known to some of you and maybe more peripherally known to others of you. And hopefully there's some people here who haven't heard of Darren and can learn about what Darren's doing. Darren is now a laboratory head at the Monash Institute for Pharmaceutical Sciences, MIPS, up the road. And he uses metabolomics and proteomics to study and understand mechanisms of drug action and drug resistance. And he does that for infectious diseases like malaria and African sleep sickness caused by a parasite, Trypanosoma brucei, which is spread by the bite of an infected fly, the tsetse fly. So we'll hear a little bit about that parasite today, in addition to malaria. Darren completed his PhD at Monash University in 2007 and then undertook several postdocs in Uganda, in Scotland, and then returned to Australia to undertake a postdoc position with Malcolm McConville, where he conducted studies on metabolomics of parasites. In the 11 years since obtaining his PhD, Darren has been really quite successful, has published over 75 papers and has obtained lots of grant funding and fellowships. And last year was awarded the top-ranked CDF2 from the NHMRC in the Biomedical Sciences category. And this year was awarded the Research Excellence Award from the NHMRC for that achievement. So we're going to hear about what Darren's doing at the moment, and without further ado, I'll welcome Darren up to the stage.
Yeah, thanks, Justin, and really thanks for the invite to come and share some of my research with you. It's a real honour to be presenting in sort of what is the home of malaria research, certainly in Melbourne, if not globally. And so a place that's probably led the way locally in terms of systems biology. So parasites and metabolomics, it's great to come and share some of our work with you. So working in metabolomics actually gives me the opportunity to work on lots of different fields and pursue lots of interesting science. But what's most close to my heart is the research on the tropical parasites and in the drug discovery space for diseases such as malaria and trypanosomiasis. So just to briefly mention, trypanosomiasis is a disease. It's restricted to Africa, as the name suggests, and it's an infection by the parasite Trypanosoma brucei, which is an infection of the blood that you can pick up from a tsetse fly. The initial infection is not too dangerous, but eventually this parasite will make its way into the brain, and then it becomes lethal. So that's a particular problem in sub-Saharan Africa. Another major issue in sub-Saharan Africa is malaria, as I'm sure you're all aware, which is spread by mosquitoes. So I'm sure you've seen the life cycle of Plasmodium before and heard some of the statistics, but just as a different way of giving a background to the problem at hand, I thought I'd share a little bit of data from some work I did when I finished my PhD. I went to Uganda for a year because I wanted to see what was really happening in the field in terms of these diseases. And we did a study on the Artemisinin combination therapies. And these are the current first-line treatment, also the last-line treatment. They're the only treatment, really, for malaria. In that study, we were looking at pharmacokinetics, so understanding concentrations of drug in the blood over time. And we're exploring this sort of unusual combination. So the artemisinin combination therapies have an artemisinin, such as dihydroartemisinin, or DHA, and a long-acting partner drug, such as piperacin. And what happens pharmacokinetically is that the artemisinin is cleared very quickly over it's a half-life of sort of 1 or 2 hours. Yet the partner drug hangs around the body for days, weeks. I think in one patient we were still detecting it 84 days after we gave them a dose. So you have this real mismatch in pharmacokinetics, which can be problematic, particularly in terms of the potential for generating resistance. Parasites like bacteria are always going to generate resistance to drugs they're exposed to, And if you expose something to a single drug over a long period of time, as is the case with piperequine, then it's only a matter of time before resistance is going to emerge. And so this study was done 10 years ago. In the last couple of years, we've now seen, at least in Southeast Asia, resistance to piperequine emerging, and it's perhaps not too surprising. One of the other issues with artemisinins, or artemisinin combination therapies, and it's actually, again, the partner drug that's often the issue, is the side effects. So actually they're fairly safe. They don't cause a lot of serious toxicity. But when we were looking at the use of these medicines in children, which is about 90% of the people suffering from malaria are very young children, well, in the case of piperequine, 15% of those children were vomiting up their dose immediately after they took it. With lumefantrine, it was only about 7%. But you can sort of see here that, okay, in theory, if the drug's given correctly, taken correctly, it works, and it works pretty much 100% of the time, at least in Africa. If the child's going to throw up that dose, they're not going to absorb it, of course, and therefore your efficacy is going to suffer. So it's worth sort of keeping in mind just what we would think is a fairly minor side effect in terms of the efficacy. At that time in Africa, and even now, the efficacy of artemisinin combination therapies is very good. It'll clear over 90% of infections. And the issue is actually re-emergence. So in that study, which was in Uganda, generally we found people were clearing an infection and they were protected for up to about 4 weeks, which is while the piperequine levels were above around 25 or 24 nanogram per mil. And then once these levels get low enough, the new infections start to come in. And in some of those populations, it was almost immediately, because there's people getting exposed to infectious bites every day. So as soon as the drug goes below a certain level, the next episode comes on. So that was the issue then. The issue we're facing now is much more serious, and that's Artemisinin resistance. So now in Southeast Asia, we're seeing lots of cases where people aren't responding well to artemisinin combination therapies, and we're getting treatment failures occurring. At the moment, that issue is limited to Southeast Asia. A lot of us think it's probably only a matter of time before it becomes a problem in Africa, and the consequences of that would be diabolical. The other issue I'll mention briefly is cost. Remembering these are developing nations where these diseases are mostly an issue. And artemisinins actually come from a plant. About 1 or 2% of the plant material can produce artemisinins, but that's quite an inefficient process and can be quite costly. Just an anecdotal sort of report, when I was in Uganda, I used to like riding around the villages in the spare time. And this is just a pharmacy that I came across in a little village, and they just sort of had 4 pots of pills sitting on the counter, and I think one was paracetamol, one was amoxicillin, one was chloroquine. So chloroquine is an anti-malarial. We know that malaria is very common here, but the malaria, or at least the parasites in this region, had been resistant to chloroquine for decades when I took this photo, yet this is still a first-line treatment that most people are getting for malaria. And I asked the pharmacist, I said, why are you giving out chloroquine? Why not artemisinins when WHO is recommending artemisinins? And she sort of turned around and picked up a dusty little box off the back shelf and dusted off and said, oh, is this what you mean? And I was like, yeah. And she's like, I can't remember what the number was, 50,000 shillings or something. The cost was just astronomical for these sort of people living in this village that just couldn't afford it. So it sort of made me think it's not just the medical side, it's also the logistics in terms of getting drugs out there that are going to be useful for people. And as one way to overcome this, we've done some work developing fully synthetic peroxide antimalarials, and I'll talk a bit about that later in the talk. So that was the malaria side of things. In terms of trypanosomiasis, similar sort of issue. We have some drugs available. They sort of work, but they're not very good. We need new ones. The first line treatment for stage 2 HAT is, or human African trypanosomiasis, is eflornithine. It's not a very potent drug, so you need to give huge doses. I think it takes 14 bags of saline to give someone a course of treatment. And remembering that most of these patients are well into the jungles in the DRC and remote places in Central Africa, it's really just not practical to get the hospital facilities that are required, let alone get the drug there. Well, the alternatives are nifurtimox and milazapine. Any chemists in the audience might sort of notice some of these features are particularly toxic. So milazapine is essentially an arsenic derivative. As you might imagine, that's particularly dangerous. interesting side note. So the arsenic derivative, or at least arsenic, was found to be a great medicine for trypanosomes in the mid-19th century when the explorers were sort of roaming Africa, and it was actually Dr. Livingstone, I presume, who was finding these horses were dying, and they were getting trypanosomiasis. Well, he didn't know what was the cause at that stage, but they were dying, and when he gave them arsenic, they were getting better. So he kind of figured out that the arsenic would kill this parasite, and many, many years later, we're kind of still at the same point. We're still using arsenic to treat people. Resistance is now becoming an issue in terms of eflornithine, at least. In the lab, we could generate resistance within a couple of months. And now it's in the field starting to become a problem. So it's now being combined with nifurtimox to get an effective treatment. And again, cost is a major barrier for these populations. So sort of after all that, I guess the moral of the story is we need new drugs to kill these parasites. And there's a few different approaches we can take to that. I guess generically speaking, drug design can take a target-based approach, or it can take a phenotypic approach. So a target-based approach is where you find an essential protein, maybe it's an enzyme, and you design or screen compounds that are going to inhibit that enzyme. I'm sure many of you are working on a similar approach here. In reality, in the antiparasitic field, this approach hasn't sort of produced a lot of new drugs. The good thing about this approach is that at least you know how the drugs are working. If you find something that kills a parasite, you know how it's killing the parasite. The alternative approach, this phenotypic screening approach, has actually worked really well in the parasitology field in terms of it just requires growing the cells in culture, growing the parasites in culture, screening large libraries of compounds against that, and finding ones that will kill the parasite. So we've, I guess as a community, many compounds have been identified that will kill malaria parasites, that will kill trypanosomes using that approach. But for a lot of these compounds, we really don't know how they act. That in itself is not a hard barrier, but it does restrict the development of these compounds if you don't know how to optimise them, if you don't know how they're working, you don't know how they should be dosed, what they should be combined with, what to look out for in terms of resistance. So a lot of my research is really using metabolomics, a technique that we specialise in, to try and understand mode of action of these compounds, mostly for the ones with unknown action, but even it's been handy sometimes looking at compounds where we think we do know the mode of action. Sometimes there's multiple targets or off-target effects that can be picked up with an omics approach that don't get picked up with a sort of traditional target approach where you're only looking at one outcome. So I think most people here should be fairly familiar with systems biology, and sort of often it's put forward this way, is that your genes lead to your transcript, lead to your proteins, and so metabolomics is really the measure of the small molecules that are the result of the reactions that the proteins, the enzymes, the transporters, these sorts of things undergo. And why it's attractive for looking at drug effects is that sort of this end of the omics cascade is much more phenotypic. This is what's actually happening to the cell or the organism at a particular time, whereas the genome is kind of a fixed genome. Adding a drug won't change the genome very rapidly. It would take long-term selection to change that. So, of course, I prefer to show this way, actually, with metabolomics clearly at the top and the most important. So for those that are not familiar with metabolism, I'll get you to memorise this. This is your biochemical pathways. Essentially, it's what metabolomics is. It's measuring all these small molecules in the cell and trying to understand how they're interacting, how they're being affected by something. So in the case of a drug action study, we'll measure the metabolome, we'll add a drug, and we'll see if some of these features increase or decrease in abundance, and that'll give us an idea of which pathway the drug's acting on, and then we can follow it up from there. The actual methodology, I'll briefly just show you for those that are interested. The extraction step, by the initial stages in the lab, they're actually not too difficult compared to a lot of other techniques. The key is really quenching metabolism very rapidly, because metabolism changes quite easily. Just leaving something out on the bench can be enough to change the metabolism for something. So it's really important to quench that quickly, and then wash down your sample, extract it in an organic solvent, and then we can run that on the mass spec. So we try and do as little as possible to the samples. The mass spec analysis, our preferred approach is using a helic chromatography based approach, which is good for separating hydrophilic molecules, which most of our central metabolites are. And we couple that to an Orbitrap, which gives high-resolution mass spec. And the advantage of that is that with high-resolution mass spec, just based on the spectra alone, you can, a lot of the time, determine the molecular formula of whatever it is you're measuring. So if you see a mass of 175.1190, like straight away we know, okay, that corresponds to the formula for arginine. And if we have libraries of metabolites to look at, then we can very quickly go from mass spectral data immediately to lists of metabolites, or at least a short list of metabolites, and try and understand something about the biological function that we're interested in. That process is probably not as easy as I just explained it then, but I've put a fair bit of work into developing software that helps with that. So the Diene software is it's a combination of packages that extracts out the raw data. So each sample will give you a bunch of peaks. It'll extract those out, align those with databases to identify a list of metabolites, and then you can look at that statistically to see what's different between groups or map them onto pathways to understand what's happening with metabolism. So that's the general approach. As a first example, just to sort of show you how it might work in terms of investigating drug action, actually this is one of the first studies we did when I was back in Glasgow in Mike Barrett's lab, setting up this whole workflow, and I was working on Trypanosoma brucei and looking at the sort of one of the first line drugs for that, which is eflornithine. So we put in eflornithine and we watched it over a time course from 1, 24, 48, 72 hours to see what happens to metabolites. And here's sort of a heat map which shows a lot of metabolites there. The yellow means it's not changing, red means it's increased in abundance, blue means it's decreased in abundance. So overall there wasn't actually a lot changing. There was only 5 things that were significantly changing. And if we zoom in to see what those 5 were, the things that were significantly going down over time were putrescine and its acetyl form. And the other end of the scale, the things that were increasing were ornithine and its acetyl form. So just in an untargeted fashion, looking, okay, we put the drug in, what happens? We can straight away see that the effect of the drug is in this polyamine synthesis pathway. We see putrescine going down, we see ornithine going up. And actually, ornithine is a substrate for ornithine decarboxylase, and putrescine is a product of that. So this data would be consistent with a drug that inhibits this enzyme. And indeed, this was a proof of concept study. We already know that eflornithine does inhibit this enzyme, so yes, it actually worked. It was perhaps a little surprising to us at first that we didn't see changes in arginine or changes in spermidine. But the good thing about metabolomics data is that you can do your initial screen and then you can dig a little bit deeper. So when we dug a bit deeper, we actually had a look for spermidine in the data and we could find it, and it was going down as you'd expect. It got filtered out in all those initial filtering processes, possibly because these error bars were pretty large. It's a particularly challenging one to detect with LCMS. But when you look in the data, it was there. The arginine actually wasn't changing. I mean, we detected it, it wasn't changing. And that we thought maybe because arginine is involved in a lot of other things. But actually, when we investigated more, we found the enzyme that is supposed to convert arginine to ornithine, even though there was one annotated in the T. brucei genome, when we studied that enzyme, it wasn't actually an arginase enzyme, it was actually another enzyme. And we can't find an arginase enzyme in the parasite, so it's quite possible that part of the pathway is just not there. So as well as understanding the drug action, we can use it as a sort of a starting point to investigate biology. And on that front, we had a look at those other metabolites, the acetyl form of ornithine. We found a gene that was putatively annotated as that enzyme, and then we're able to confirm that could exchange, or it could acetylate ornithine and vice versa. Likewise, we feel that the acetyl putrescine is just an acetylated form of the putrescine. We haven't found the particular enzyme involved in that, mostly because there was a few alternatives and we haven't actually followed it up. But we now know a lot more about this pathway in that parasite, just from that sort of one-off experiment, obviously with a few follow-ups. So it's that was a proof of concept to show the idea works. We then sort of moved into looking at malaria. And sort of one of the bigger projects I did when I come back to Australia, working with Malcolm McConville and Stuart Ralph at the Bio21 Institute, was to investigate this thing called the MMV malaria box. So basically what this is, it's a collection of compounds that have come out of screens from these companies and research institutes. So some of those screens identified over 30,000 compounds that could inhibit the parasite or novel antimalarials, but we didn't know the target. So we thought, let's try and investigate the target with metabolomics, and rather than look at 30,000, MMV, the organisation, produced what they call the malaria box, which was a subset of these compounds. So we had a look at those, or at least 100 of those compounds initially. But to start with, we used 10 of the known sort of antimalarial compounds just to see how the metabolome of the malaria parasite was looking. And here's some data, which probably doesn't mean a lot, but it just sort of shows that we get some clustering. So here's chloroquine and piperacin, some of the common artemisinin partner drugs. Just by doing a hierarchical clustering analysis, we could see the replicates of those clustered together, and they were having the same sort of effect on the parasite. Likewise, artemisinin and methylene blue, both antimalarials that act by redox processes, they have sort of similar effects on the parasite, and atovaquone also clustered well. And actually, these other compounds that didn't cluster in any particular fashion, when I look back later, I realised that they're actually not very potent antimalarials, and the concentration I used, 1 micromolar, was below the IC50 for those. So we really didn't expect a response for those, and indeed, we didn't get a metabolic response for those. So again, when we're getting an effect of the drug, we're getting a metabolic profile. And probably one of the classic ones that I'll show you initially was atovaquone. So it's a currently marketed antimalarial, probably more used for people travelling to these areas than for people living there. But that had a very specific effect on metabolism. So there's a couple of metabolites increased, a couple decreased, most metabolites not affected. And if we look at what the metabolites were, as they're shown here, for those that did remember all your metabolic pathways from undergrad, this is the pyrimidine biosynthesis pathway. So all these metabolites are in this pathway, and it sort of would make sense that we've got accumulation upstream of this DHODH enzyme and depletion of UMP, which is downstream of that enzyme. So it indicates that the atovaquone's inhibiting somewhere in here. And actually, again, this is a drug that's been studied before, and it's known that it doesn't inhibit that enzyme directly. It inhibits this cytochrome BC1 complex, which is responsible for producing ubiquinone, and the only known role, or the only essential role of ubiquinone in this stage of the parasite is the reducing factor for DHODH. So therefore, that's why we get the effect on this pathway. So this sort of shows that we don't always get identify the exact enzyme, but we see the pathway that's involved and where the blockage is going to occur if we've got enzyme inhibition involved. So that sort of gave us enough promise to move on. We then screened 100 compounds and got data that looks something like this. I guess the good thing about this is there's a lot of yellow. Yellow is no change on this scale. So we haven't sort of put in enough drug to just kill the parasites and completely disrupt everything. But we do have some specific red and blue spots that indicate the specific effects on parasite metabolic pathways. And you see a lot of the changes were actually in the nucleotide and peptide region of the metabolome. And if we start with the nucleotides, actually the two that were upregulated by a lot of compounds, so here all the compounds tested are along the x-axis, and the y-axis shows the intensity of the metabolite peak in the LCMS, or essentially the abundance of that metabolite. So for a lot of these compounds, actually a quarter of the compounds we tested, they caused an enrichment of these two metabolites. And some of you may remember they're the two metabolites that were elevated after exposure to atovaquone. So what we actually found is that a lot of these new drugs of unknown function are actually doing the same thing that atovaquone does, which we already had well established. To break it down a little bit more, they didn't all do the same as atovaquone. Some of them have the same effect as atovaquone on BC1. Some of them directly inhibit DHODH, that other enzyme in the pathway, and they give the same metabolic effect. So Charlie Chua, PhD student working with Mal at the time, did some follow-up studies to show that in some mutant parasites, they're resistant to DHODH inhibitors. They're also resistant to these compounds as well. So just to confirm that the activity is associated with this metabolic phenotype, and it's not just some side effect of what's happening in the parasite. So that was kind of interesting, but somewhat dispiriting to see that all these new compounds are actually not doing anything new at all. And in a similar fashion, we had a cluster of compounds that had a certain signature on peptide metabolism. I'm yet to figure out what the significance of these peptides is. I don't know why these compounds increase the abundance of these peptides, but they all had the same effect, and they had the same effect as piperacin and chloroquine. And if you look at the structures, you can see some of these are very similar to chloroquine. So it's not really surprising if you look at the structures that they had the same effect on the parasite. So this allowed us to cluster some more compounds that go along with known drugs. And perhaps they're probably not good avenues for future drug discovery. And the other approach we can use with a data set like this is to look at things with a multivariate approach. So if we do a PCA analysis, for example, actually the first two components showed a lot of scatter in the data, which It's probably not unexpected in a large data set like this. You're just going to get natural variation occurring. But in the third principal component, we started to see some clusters of replicates coming out as separating from the bunch. And actually some of these compounds included one of our controls, which was artemisinin. So we have a bunch of compounds here that have a similar effect to artemisinin on the parasite metabolome at a sort of multivariate level. So similar effect, what is that effect? If I looked at the most extreme, and I'm just denoting them by colour here, but let's say the red one and the blue one, the most extreme cases, there was significant downregulation of about 50 metabolites, which covered a whole range of pathways. So it's not a specific, oh, it's inhibiting pyrimidine synthesis or peptide metabolism. It was a whole lot of things being affected by these drugs. And likewise with the artemisinins, they're very fast killers. So in this little experiment here, where I've tried not to kill the cells too much, I think these compounds, very potent, very fast, have actually just effectively stopped the metabolite, stopped the parasite from metabolizing. Certainly those four stood out. The only metabolite that increased in abundance was glycerol phosphate or dihydroxyacetone phosphate. Everything else went down. She was sort of looking at this in a separate project, but the reason this actually goes up is not because the parasite's producing more of it. It's actually because the red cells, the uninfected red cells, normally produce a lot of this metabolite. But when you have infected parasites in there, that affects the glycolysis in the uninfected red cells, and it means that they produce less. So again, this sort of shows these four compounds who have effectively killed the parasites or stopped the metabolites stopped the metabolism of the parasites, and therefore the host red cells are actually just upregulating their metabolism back to what they would normally, how they would normally be. And the other compounds that clustered in that PCA sort of cluster didn't have as dramatic effect on all these metabolites, but certainly the peptide metabolism was downregulated across the board. And these peptides that are affected here are not the same as the peptides we saw with chloroquine. These ones all come from haemoglobin. So for those people not working in the malaria area, I know a lot of you here are, the malaria parasite essentially eats haemoglobin as a source of amino acids and as a way to make space within the host red cell. And so what these compounds are doing are essentially stopping that haemoglobin degradation, so we're not getting these peptides that come out of that haemoglobin degradation pathway. I guess I can add a footnote to this is that the compounds that fell into this category, even though I say they're sort of artemisinin-like because they matched in this profile, structurally they're very different. We knew they don't act in by the same mechanism as artemisinin, but they were structurally similar to each other and to the known inhibitors of a sodium channel, which is called PF-ATP4, which is also a very promising drug target. So actually, it looks like they're all inhibitors of PF-ATP4, and the group, Kieran Kirk's group in Canberra, have done some different studies that also support that idea that these are all inhibiting that same transporter. So we sort of found that a lot of compounds fell into clusters of it looks like atovaquone, or it looks like chloroquine, or it looks like artemisinin and the PF-ATP4 inhibitors. We also found a few unique things coming up. This data's a little bit messy, but this is a particular metabolite in the isoprenoid biosynthesis pathway. And looking at all the compounds, there's one compound that we'll just call D6 that significantly depleted the levels of this metabolite. There were four other compounds that had a similar effect, and these are the four that sort of affected everything, so we're ignoring those in terms of a specific mechanism. But what it would indicate is that this compound must be inhibiting this isoprenoid biosynthesis pathway. We're measuring essentially the last metabolite in that pathway, so it could be inhibiting this enzyme or somewhere upstream of that. And at the same time we were doing this work, other people were taking non-metabolomic but very different approaches to investigate these malaria box compounds. And actually 3 other groups all independently came up with evidence that shows that the compound inhibits ISPD, this middle enzyme of that pathway. So again, there's very, very solid evidence that this particular compound is an inhibitor of that pathway. And perhaps my favourite metabolite that sort of popped out of this screen, which I sort of ignored at first. When you do an omics study, you often get sort of lots of noise in the data and you sort of see some things that don't make sense and you ignore those for a while. But I couldn't ignore this one, traumatic acid. I had to look it up, find out if it was legit and in the parasite. And sure enough, it is. So there's one compound, C7, that really dramatically decreases the level of this metabolite. The other compounds don't do it. And actually, Joanna Daly's group in New York, about 5 or 6 years ago, have already shown that traumatic acid is produced by malaria parasites. This is a pathway that's been well described in plants. It's not a mammalian pathway, so we sort of maybe didn't expect it, but parasites do have a lot of features that are in common with plants, and this pathway is one of them. So they do have this pathway. Again, we don't know which enzyme in the production of this metabolite is being inhibited, but that's a subject of further investigation in our lab at the moment. So to sort of summarise that bit of work, we screened 100 compounds against malaria parasite with metabolomics. About a quarter of them have a similar effect as atovaquone or DSM, which is the DHODH inhibitor. Quite a few have this artemisinin-like or PFA-4-like action, and there were some quinoline-like compounds. And we got a few novel targets coming up, like the traumatic acid, for example. A lot of the compounds didn't give a specific metabolome. Some of them, probably because they're not very active, like I said with that first study, the IC50s are up around the range or even higher than the amount of drug we're adding to the parasites in the screen. And some of them probably just don't act by mechanisms that affect metabolism. There's lots of other targets you can imagine if something affects the structure or some other transporters or other stages of the parasite that we weren't looking at, that we wouldn't have picked those up. But overall, that's sort of not a bad return for an untargeted approach. So to sort of finish off, I'll just want to focus in a bit more on a specific class of antimalarials that I've spent a lot of time looking at, and this is the ozonide antimalarials. So I mentioned that artemisinin is sort of first-line treatment at the moment, very, very important antimalarial, and it comes from this plant Artemisia annua. And the key pharmacophore in artemisinin is this peroxide group here. We know that peroxide is essential for activity. Without it, it has no activity. And so some chemists in Nebraska that we collaborate with, Jonathan Venestrom, came up with the idea that if we could make synthetic peroxides, we can make antimalarials very cheaply, and we can alter their properties to make them have better pharmaceutical properties, such as better solubility, longer half-life, those sorts of things. And the first sort of candidate they came up with was this one, OZ277, and that's had quite good activity in preclinical studies, went into the clinic and still had good activity, but actually when they went from animals to humans, they found the half-life wasn't sort of as good as they hoped, but more importantly, when they went from healthy people, healthy volunteers, into infected patients, the half-life dropped again. And we since figured out that it's because this peroxide group is quite reactive with iron. The reactivity with iron is actually important for its antimalarial activity, but if it's also reacting with free iron that's floating around in our blood, that'll degrade the compound as it's exposed to that. And so that's what we found, is the infected patients had more free iron in their blood. They were degrading this compound. Half-life was, I think, up around 4 hours in patients, so better than DHA, but not the sort of half a day that we were looking for. However, having done a lot of those studies, that was back in my PhD, very sort of focused, targeted studies back in those days, we sort of looked at the chemistry and the structure and figured out that if we could protect this peroxide group sterically, then we could slow down that reaction with free iron and therefore increase the half-life. And so what that led to is the second generation OZ, so OZ439 is the lead candidate at the moment. It's got this aromatic group here at this 5' position, which essentially changes the stereochemistry of this, changes the conformation of this cyclohexyl ring and protects that peroxide bond. So it's now a really stable compound. It's got a half-life of about a day rather than 4 hours. So much better exposure in patients. So we know it's promising antimalarial, but again, mechanism of action is not really well defined. We know that in the parasite, as I said before, the parasites eat hemoglobin and they produce amino acids out of that, with some peptides being produced along the way. As a sort of a byproduct of hemoglobin digestion, they produce a lot of free heme, and free heme is a redox active molecule. It's potentially very toxic. And so the parasite has a neat way to crystallise that into hemozoin, which is non-toxic. But there's always going to be a little bit of free heme in the parasite, and it's that heme which reacts with the peroxide bond of the ozonides or artemisinin to produce some free radicals. And then it's thought these free radicals kill the parasite either by interacting with proteins, small molecules, or lipids. And there's a bit of work being done now into proteins, but a lot of this is based on sort of in vitro correlative studies. We want to investigate in the actual parasite what's actually being affected, why is the parasite dying, and what are the biochemical pathways that are being affected. So, of course, metabolomics is good for this because we're looking at lots of pathways at once. We did some metabolomics, identified a lot of metabolites, and the ones that were really changing after drug treatment were peptides, whether it be the first or second generation OZ, or even the artemisinins had very similar effect. They mostly affect peptides. The peptides that were affected were all pretty much all downregulated, so it's a volcano plot there showing downregulation of peptides by the OZs. The artemisinins had an even greater effect. So this is just after 3-hour treatment, we get really significant depletion of hemoglobin-derived peptides after artemisinin treatment, and a bit less of an effect with the OZs. The peptides, as far as, at least in terms of what we can tell, are all coming from hemoglobin. So perhaps not too surprising, the parasite chews up a lot of this hemoglobin. And we've, at least those that we've done MSMS on, we can confirm that they're from either the alpha or beta chain of hemoglobin. And to sort of look at the profiles over time, I'll flick all these up, you can sort of see the red line's artemisinin, so a lot of these peptides really go down within an hour and a half of artemisinin or DHA treatment. Whereas for the ozonides, it's a bit of a slower effect. And if we extend this out, I haven't got the data here, but if you go out to 6 or 9 hours, then these curves will track down as well. But generally speaking, it's the same sort of effect, but a slower effect for the ozonides. And that agrees with what we know about the activity. So this is data from Leanne Tilley's lab that Tuo produced, where they looked at treating parasites for short pulses with these different drugs. And if you focus on the blue line, that's a 3-hour pulse. The artemisinin in 3 hours gives a full kill up at around a micromolar concentration, whereas the same concentrations of the OZs don't really kill many of the parasites. If you extend the duration up to 6 hours, then all 3 of the drugs give good killing effect. And interesting, the metabolomics data, over 6 hours we do see those hemoglobin peptides coming down with the OZs, and we also see some other pathways starting to be involved as well, some nucleotide synthesis and some lipid biosynthesis pathways. So it's likely that the OZs do have sort of these additional effects that bring on the slightly slower activity. Even though it's slower than DHA, I will say it's still quite fast compared to a lot of the other antimalarials out on the market. Given that peptides are interesting, we wanted to have a look at some of the other peptides because in the metabolomics analysis, partly because of the way we prepare the samples, partly because of the way we analyse the data, we can measure things up to about 4 amino acids in length quite easily. You can go a little bit higher, but when you're degrading hemoglobin, you're producing a lot of longer peptides in intermediate steps. And we're not getting these in the metabolomics data, so we sort of developed a peptidomic approach where it's more like a proteomics workflow, but without a trypsin digest step. And then we sort of ran that to identify peptides and found a bunch of plasmodium and human peptides. And just presenting these in a sort of a mirror plot. So here's our sort of loading control. HSP70 is commonly used for that. We can see there's sort of 2 peptides identified from that protein. Levels in drug treated are the same as levels in control if you look at the height above and below the line. Whereas where we saw differences were actually all the hemoglobin-derived peptides. So after drug treatment, a lot of these longer peptides were actually elevated compared to control. So this is actually the opposite that we saw with the short peptides. So given it's a multi-step process to degrade hemoglobin, there seems to be a blockage sort of halfway along that process where the large peptides are being increased and they're not being converted to those small peptides. So it indicates either these proteases here specifically being interfered with or the environment of the food vacuole is such that it doesn't promote that degradation. This is consistent with some other work that's been shown that some of the protein targets of ozonides, when people have done pull downs with these, they've identified particularly plasmepsins and other proteins in the digestive vacuole. So it is sort of consistent with that. We did some activity-based protease probe analysis and some proteomics to see if we could see any changes in activity of the cysteine proteases. Actually, all these proteases that we could measure were upregulated. So it kind of, again, consistent that if you've got an inhibition of the pathway, the parasite's responding by upregulating the production of these proteases. So that sort of indicates that does seem to be the main initial target of these compounds, both the OZs and the artemisinins. And just to sort of finish off with, talk a little bit about artemisinin resistance, because understanding drug mechanisms is important, but what's really critical now is to understand how that sort of how that interplay relates with the resistance that we're seeing now in Southeast Asia. There has actually been a case reported recently in Africa of artemisinin resistance, and it's really a potential disaster if we get widespread artemisinin resistance in Africa. A lot of the successes of malaria control over the last sort of 15 years could be reversed very quickly. So what do we know about artemisinin resistance? Well, some genomic studies, some population genomic studies, and also some lab-based studies have very clearly shown that this protein here, Kelch13, or K13 I'll refer to it as, is mutated in a lot of the parasites that are associated with artemisinin resistance. When I say it's mutated, it's usually a single point mutation, but there's been, I think, over 100 now different single point mutations in this protein identified that are associated with resistance. So essentially mutating, I'll say anywhere, it's probably not anywhere, but a lot of places in this propeller domain can lead to a resistance or a lesser effect of the artemisinins in the parasite. How that plays out in the field is a prolonged parasite clearance. So where we would have seen clearance of the parasites within 3 days, now it might take 4 or 5 days to clear the parasites because they're more resistant to the drug. So we wanted to investigate this, and we were using some clones that David Fiddick's lab produced where they took an isolate from Cambodia, from the field that was resistant and had this R539 mutation in its K13, and they also reverted that in the lab to the wild type form, and that turned the resistant parasite into a sensitive one. And they also made a 580Y mutation, which is another very common resistance mutation in the field, and showed that that was resistant. And we've also done some work on some other field isolates from the sensitive and resistant camp. And our approach here was, we want to know overall, what is this K13 doing to the parasite? So we went with the multi-omic approach, trying to figure out how many ways we could use our mass spec. We did metabolomics, we did proteomics, which we split into the native peptides and also the digested proteins to give us a full proteomic analysis. In the proteomics, when we first look at rings, rings are a little bit hard to work with, so the data's a bit more variable than I would have liked. We got a few proteins that were significantly different between the resistant and sensitive lines. One that sort of stands out is the actual mutated protein itself is lower in abundance in the resistant parasite. We then had a look at the trophozoite stage, so the older stage of the parasite, which is actually a bit easy to work with. You can get cleaner data. We showed that the resistance can be observed in these older parasites, even though the biggest difference in drug sensitivity is in the ring stages. If you just pulse for a short time, like the short half-life we see in the field, then you do get a difference in sensitivity for the troph stage of the parasite as well. And looking across a bunch of different lines, we always see that this K13 protein is lower in abundance in the resistant parasite compared to the control. So we're able to show that it's not just a mutation that's associated with resistance, it's actually the abundance of that protein that is affected by the mutation and is associated with resistance. And we did that with some dimethyl labeling proteomics just to make sure we've got quantitative differences, which I think was clearly shown. But the proteomics actually didn't show a lot else. We were hoping to see a whole lot of other pathways regulated by this K13 mutation, and sort of a lot of things have been proposed, but actually nothing came up. Once you do a few replicates and iron out all the noise, there's not a lot of change in the baseline levels of these pathways. But we had a look at metabolome as well, and indeed we could see some differences in metabolome between this is the sensitive line and this is the resistant lines. If we sort of look at what metabolites are associated with that difference, there was again very few. We didn't see wholesale changes in metabolism. But one thing that did stand out was glutathione and its precursor gamma-glutamyl cysteine. They're at higher levels in the resistant parasites, about 2-fold higher than they are in the sensitive parasites. So this kind of would fit in with what we know about artemisinin activity. We know that hemoglobin is digested to produce heme, and that heme reacts with artemisinins or ozonides to produce free radicals, and then that's associated with damage to the parasite. So if we have a resistant parasite that's got more glutathione, you'd expect that glutathione to be able to mop up a lot of those damaged, or either the radicals themselves or the damaged proteins that result from that. So that sort of might explain partly why these parasites are resistant. The peptidomics analysis was also somewhat interesting. Again, looking at the hemoglobin-derived peptides, this time we saw the levels of these hemoglobin-derived peptides were higher in the sensitive line than in the resistant line, which sort of indicates, and that's in both the forms of hemoglobin, so it indicates that the conversion of hemoglobin, or the degradation of hemoglobin, is less in the resistant line. Other studies have shown that if you inhibit these or knock out these falcipains, again Leanne Tilley's group have shown quite nicely that this falcipain activity is important for artemisinin activity. If you knock that out, then you just block the activity. So if this whole pathway is just downregulated, that might explain some of the less effect of the drug in the resistant parasites. Also with proteomics and protease probes, we could see that the protease activity and the level of the falcipains and DPAP1 and other cysteine protease, they were all lower in the resistant parasites, indicating a small but significant downregulation in that pathway. And I won't go through the animation. So in summary, we've now sort of got a reasonable understanding that hemoglobin-derived heme is important for activating artemisinin, produces free radicals, which initially hit targets in the food vacuole, but I'm sure go on and hit other targets as well over time. Normally, the parasite's main response is through glutathione, and that can mop up some of those radicals. But in a resistant parasite, you've got less of this hemoglobin degradation, so probably less production of these artemisinin-derived radicals, and you've got more glutathione production, so that can mop up the radicals and help to delay the effect of the drug. Happy parasite. Sad patient. So with that, I'd just like to thank everyone that's been involved in this work. So my team, particularly Giselle and Carlo, over there hiding, that have done a lot of the work shown here. Also Sue Charman worked with a lot on the OZ project, and Laura for the protease work. Mal and Stuart and Leanne from Bio21 for involvement in a lot of these projects, and also Mike Barrett, who's very significant in helping us sort of get this metabolomics approach up and running and applied in these scenarios, and of course the funding agency as well. Thanks.
Thanks very much, Darren. Great seminar. Thank you very much for that. We've got plenty of time for questions. I think that was a really nice overview of the utility of metabolomics for lots of different areas of research. And I think one of the greatest questions in malaria at the moment is around drug resistance, because drugs are our frontline defence at the moment. So understanding resistance to artemisinin, for which the Nobel Prize was awarded in 2015, was one of the biggest questions, I think, in the drug resistance field. So it's a very perplexing area. So thanks again, Darren, for that. So if anybody got any questions for Darren? Chris?
Darren, you see all these changes in hemoglobin degradation. How sure are you that that's some kind of direct effect, or it's not some downstream effect of the parasites dying?
Yeah, it's
slowing down their metabolism or their ability to degrade their food source.
Yeah, it could be. I guess the good thing about the omics approach is that we're seeing when we're seeing effects on hemoglobin metabolism, the other pathways are not affected. They're still making peptides, pushing through glycolysis, making lipids, all that sort of stuff's not affected at the same time hemoglobin degradation goes right down. Essentially, these peptides are disappearing. So that gives us a sense that it's an early effect. And actually, with probably best effect, with the PFAW4 inhibitors, you can hit them with drug within an hour or so, you can block hemoglobin degradation, but then if you take the drug off, they'll come back. So they're not dead, but they're affected, like so they've stopped. But that's a case where it's not hitting the enzymes in hemoglobin degradation, it's affecting iron homeostasis, and that must affect either the pH of the food vacuole or osmolarity, or in some way affect the activity of those proteases.
So the amount of free heme is probably important, as you've said, for the activation of artemisinin. But do you think that there must be some other effect that artemisinin is having on the degradation process itself? What do you think that is?
Yeah, jury's out on that. It's a lot of things, and we need to do some more work on this, but are pointing to actually the uptake of hemoglobin. So it might not actually be the degradation of hemoglobin that's being affected. I think actually Leanne's lab quite a few years ago showed that artemisinins do decrease hemoglobin uptake, or at least that endocytosis effect. So it actually could be that. It might not be the degradation process itself. It's just really hard to get solid evidence of that at the moment.
So there's been a couple of papers showing that artemisinins can kill certain leukemia and multiple myeloma cell lines in even patient-derived samples. Have you or somebody else done proteomic or metabolomic assays to see what changes happen in these cells when they're treated with this compound?
I haven't. I don't think anyone else has, actually. Yeah, a lot of people have looked at the general effect, and there's been a lot of targeted studies on various pathways and whether they induce apoptosis and by which mechanism, but I don't know of any metabolomic or proteomic studies on those.
In the box experiments at the front, there was one of them which had an effect on peptides, but not the artemisinin. It looked like a lot of them were dipeptides, but is that just because you didn't do the full-length peptide analysis? And if they really are just dipeptides, is that because it's having an effect on a dipeptidyl peptidase or something like that?
Yeah, good question. So we haven't really followed it up. So yeah, if we haven't looked at the long peptides. I should check whether we might have included chloroquine as a control in one of our other studies. I should actually look at that. It hasn't been a focus of us. There are some, I don't know if they're on that list, but there's some tri and tetrapeptides that do seem to be affected as well. But yeah, you're right, they're mostly dipeptides, but we don't know where they're coming from or why. Problem with dipeptides, they're so short you can't align them to a protein very easily because they'll align to everything.
And then why not?
I was interested in the differential pharmacokinetics of the drug that you were testing in uninfected versus infected people. And is that general? I mean, does that happen in all of these drug screening tests? And have you tested in animal models to see if the same effect is true in uninfected versus infected?
Yeah, so it's not unheard of before, but it's quite specific to this particular series because they have that peroxide bridge that's a stability liability, and it's, again, it's unusual that a chemical stability is the main route of clearance of a drug. Most drugs are metabolised hepatically. So, I mean, there are, I guess the more common case would be where an infection affects liver function and then the liver's metabolising things faster or slower, so that certainly can have an effect. But yeah, it's an unusual case, and it was only by looking at the mechanism that we could explain it.
So how, with the documented case for the African Artemisinin Resistance, how different is that from what's occurring in the Greater Mekong region? And
Yeah, from memory, it's a while since I looked at that. I think it was in Equatorial Guinea, so it was in quite an isolated environment. And it was a K13 mutant, I think, from memory. So there's some similarity there. I think there's some probably demographic aspects of Equatorial Guinea that, in terms of how they use drugs and what drugs are available, that might actually have a lot more in common with the way drugs are used in Southeast Asia than across other parts of Africa. So there's probably a lot of factors at play. I wouldn't say that one case means that in 2 years' time it'll be spread across Africa, but it's only a matter of time. Just how long?
I might ask a quick one. So notwithstanding the importance of K13, I think there's evidence that there are other targets. I'm wondering if you're doing anything in that space as well with respect to Artemisinin resistance.
Yeah, so not directly. We've just been talking to some of the main groups in Bangkok that are running a lot of these studies, and they will send us some parasites soon, but I think they're all K13 mutant. It's sort of got to the point now where the 580Y mutation has kind of become widespread across Southeast Asia, and it's kind of become the dominant mutant, and that's the main issue. But interesting you mentioned that. There was a report, I think last year, of a clinical case, Artemisinin resistance, not a K13 mutant, but they did detect differences in thiol levels. So glutathione seemed to be involved, or at least redox capacity seemed to be involved in that case, even though it wasn't a K13 mediated resistance. So it's sort of, they could all be sort of coming or linking back to that sort of redox stress response issue.
Okay, if there's no further questions, then let's thank Darren for a great talk.